Gentlemen and ladies, welcome to a continuation of pharmacokinetics basics, and this part concludes the principles about drug distribution and the factors that influence it. So in terms of drug binding to cell and tissue constituents as a factor that affects drug distribution, you find that some drugs as chemical substances, they are able to react or interact chemically with tissue and cellular components. And in this case, you find, for example, that chloroquine is highly concentrated in the liver because the ability to interact with hepatocytes. Tetracycline deposits a lot in bone tissue or osseous tissue because of the ability of tetracycline or the tendency of tetracycline to react with calcium and complex with it. Also, iodine tends to be concentrated more in salivary glands as well as thyroid glands because of its affinity for thyroid tissue. Therefore, you find if you administer iodine in thyroid conditions, it will be useful because it is highly concentrated there and it will affect how thyroxine is processed. So that is very, very important. We begin to look at metabolism as the third component after absorption, distribution, and metabolism now in the processes or components of kinetics. And that as the drug gets to be distributed, sometimes it passes through tissues that are able to change the chemical structure of these chemical substances called drugs. And in this case, because these are biological systems, sometimes drug metabolism is referred to as biotransformation. And so, as a result of these changes that take place that affect the drug in terms of its chemical structure, you find that this can lead to abolishment of the activity of a drug. Sometimes the drug may not be neutralized. It may actually be promoted in terms of activity. For example, we have drugs like prednisone, which is converted to prednisolone in the body and then becomes more active. Sometimes it's not just neutralization of the drug after metabolism or increasing its activity. Sometimes the drug metabolic reactions tend to change the active drug of the drug to another active drug. We have examples of how codeine is in the body converted to morphine. And so you begin to see the pharmacological effects continuing while codeine is in the body after metabolism. So this is very, very crucial. It is also important to realize that after you've been able to learn this part of pharmacokinetics, you need to be able to describe the different types of drug metabolic reactions that take place in the body. So the types of metabolism or biotransformation reactions that occur, we have the non-synthetic and synthetic ones. Non-synthetic because mostly these are oxidation, reduction, hydrolysis kind of reactions that take place. And so these type of reactions, the non-synthetic, are also called phase 1 reactions. Why? Because they tend to make the drug more reactive to the next phase of reactions, okay, which is the synthetic one, where another moiety or another chemical substance is added to the chemical structure of the drug, and that tends to change its chemical nature into a conjugate. So this process is phase 2 reactions called conjugation reactions because there are other chemical substances as additional moieties or components that are added to the drug. And so the phase 1 reactions, they convert the parent drug to a more polar or metabolite which is excreted or becomes more liable to subsequent reactions, which are the conjugation reactions. While a phase 2 reaction tends to make or form a highly polar and rapidly eliminated conjugate. Usually the phase 2 reactions are actually always inactive. Then the types of enzymes involved in metabolic or reactions of drugs, you find what are called microsomal and non-microsomal enzymes. So when enzymes are harvested from cells, you find that some of the enzymes will be those that are found in the endoplasmic reticulum. So they are harvested as microsomes. So such enzymes are called microsomal enzymes. Nothing special about them. Other than that, the rest of the enzymes will be those that will be found free in the cytoplasm, and so they will be also able to cause drug metabolism. In terms of factors affecting drug metabolism or biotransformation, we find that species variations may take place, and then you may find that different a particular enzyme may have different types and the reactivity of such enzymes. So the same enzyme may be different in different people. Also, the age, extremes of age. For example, the very young, you find that their liver, which is the main metabolizing organ, is still developing, and so the enzyme systems are not fully developed, so their metabolic capacity reduces compared also to the adult young man, which is the standard in physiological sciences. Also, the extremes of age, like the old, those above 60, and those also the malnourished, you find that the capacity to metabolize goes down because the liver also has become compromised, and so age in that sense tends to affect drug metabolism. Sex, you find that the gender, in females we have a lot of estrogen compared to males. Males have a lot of testosterone, which tends to increase the metabolic capacity, so the basal metabolic rate is higher in males because of testosterone. But estrogen tends to be inhibitory compared to testosterone, and so you find the metabolic capacity of females is less. Some diseases, in this case pathological factors, some diseases tend to reduce metabolizing capacity of individuals, such that you find that liver disease, for example, have a depression in the ability of someone who is supposed to be having normal metabolizing capacity. It is reduced as a result of disease. And these diseases can result from so many causes: infections, alcoholism, and viral, bacterial, food stuffs like exposure to aflatoxins, so many things. The metabolism of some drugs is reduced in cases of heart failure because the delivery of the blood and its constituents, including the drug, is reduced when there is heart failure. Also, shock, which is circulatory capacity reduction in individuals, can reduce hepatic blood flow, leading to reduced delivery of the drug to metabolizing organs like the liver. Environmental factors actually are survival mechanisms for the human race. So if you are exposed to pesticides, for example, that induces the liver to increase its metabolic capacity. Also, those people that are exposed to smoke, there is increased metabolizing capacity of the liver. Number 6 is where drugs can actually stimulate and others can inhibit. So there are certain instances where we say we cannot give, for example, this drug and the other one. Why? Because they may affect how they each of these two drugs will be metabolized by the liver. Some drugs are enzyme inhibitors, while others are enzyme inducers. So in terms of enzyme inducers, in enzyme induction, we are talking about certain drugs which increase microsomal activity of enzymes, which leads to increased metabolism. We have examples of enzyme inducers like phenytoin, carbamazepine, phenobarbital. These are used in convulsive disorders like epilepsy. We also have rifampicin. We have nicotine as well from beverages and the others, even smoke. So all these, they increase the activity of liver enzymes, leading to increased metabolism of drugs in the body. Therefore, if drugs that are metabolized by those microsomal enzymes are given together with phenytoin, carbamazepine, and all these, you find that their duration of action will be reduced, their effectiveness will also be reduced. So enzyme induction is reversible because the moment the drug that induces metabolism is removed, then you tend to have a reversal of such effects. The importance of enzyme induction, I've mentioned some of it, is that it is the cause of some of the drug interactions. Tolerance also, you find that when the enzyme induction is occurring, the amount or the dose needed to produce a pharmacological effect increases. So that is tolerance. Also in therapy, sometimes enzyme induction is used in therapy, for example, where you have hyperbilirubinemia, which you want to increase in terms of which we want to clear by increasing conjugation. You find that if we give phenobarbital, it will increase bilirubin conjugation, and this can actually help to reverse the situation. Also, enzyme induction is a mechanism to adapt to environmental pollutants, like I mentioned. So those who are exposed to smoke, those who are exposed to pesticides, their livers tend to increase in terms of metabolism to actually help defend the body by metabolizing the toxins fast. Enzyme inhibition, this is a reduction in enzyme activity, leading to reduced pharmacological effects of such drugs that are metabolized by such enzyme systems. Examples, we have chloramphenicol, erythromycin, ciprofloxacin, cimetidine. We have ketoconazole for fungal infections, monoamine oxidase inhibitors for depression and such conditions. Drug elimination is very, very important, but we want to mention that the total elimination or removal of a drug from the body is a function of so many organs. So we have the respiratory system working, the liver working, we also have the kidneys, but the kidneys are the major eliminating organs of the body. So elimination of a drug from the body may involve processes occurring in the kidney, liver, lungs, and other organs. So for us to calculate clearance, we have to find the rate of elimination divided by the drug concentration. So what is drug clearance? Drug clearance basically is the volume of the fluid from which all the drug is removed per unit time. In other words, we say it's the measure of the body's ability to eliminate drugs. So if you are defining drug clearance, you mention it's the volume of fluid. Remember the volume of distribution? Now, out of that volume of distribution, how much of it is completely cleared of the drug per unit time? And so the two major processes by which elimination takes place is by hepatic metabolism through the biliary circulation, and also renal filtration through secretion as well as a process. So total body clearance is the sum of all these clearances happening in all these organs. What are the factors that will influence drug clearance? The blood flow is important, binding of drug to plasma proteins. So the more the drug is bound, the less clearance. Activity of processes responsible for drug removal as hepatic enzymes, glomerular filtration. So when all these processes are very active, you find that drug clearance will be also very high. Hepatic clearance is a function of blood flow and extraction ratio. So the more blood flow happens to the liver, the more the drug is cleared. And so you can have all these calculations, and in the end we have to determine the half-life, which is a derived function parameter of metabolism and clearance. And so the last thing is you should be able to calculate half-life, and we'll also relate it to volume of distribution, we'll relate it to clearance and all those, so that we can connect. Thank you very much. Bye-bye.